Hello and welcome to Trailblazers. This is a series of conversations with Columbia University faculty whose ideas shape our world. I'm your host, Dustin Foote, and today we're talking about tissue engineering and the future of medicine with Professor Gordana Vunjak-Novakovic. Professor Vunjak-Novakovic is a professor of biomedical engineering and one of the world's leading researchers in regenerative medicine. Her work focuses on engineering functional human tissues to study disease, test therapies, and repair damaged organs. From lab-grown tissues to personalized medicine, her research bridges biology, engineering, and clinical care. Today we're talking with Professor Vunjak-Novakovic about her work and how advances in tissue engineering could transform how we understand disease and treat patients. Professor Vunjak-Novakovic, welcome to Trailblazers.
Thank you. It's really a pleasure to speak with you.
So we're here in Columbia's Medical Center. Can you tell us a little bit about what goes on here in your lab?
We are really trying to tackle some of the greatest challenges of our time, meaning that we are trying to develop better diagnostics. We are trying to address some of the most devastating and currently incurable diseases. A lot of research in tissue engineering, not only in my lab, but actually everywhere around here, is focused on diseases such as cardiovascular and immune and cancer. And these are diseases that really, unfortunately, take more lives than any other ones, still without definitive solutions.
Yeah, before this conversation, I also saw a video about your lab engineering jawbone. Could you tell us a bit about that?
Sure. So the jawbone was actually one of the first tissues that we have grown here at Columbia after I came here. And the project started with my very first postdoc that came to the lab after my move from Boston. Warren Grayson, he is today very successful faculty at Johns Hopkins University. He really wanted to do something about bone tissue engineering, which actually my old lab was studying for about 10 years already. And he wanted to do something very challenging, something really complex. So we thought about what could be the most complex bone in the human body to be engineered. And we decided to attempt to engineer the jawbone for two reasons at least. One is this is anatomically or geometrically very complex bone, which is very difficult to make in this exact shape. We also decided to make this jawbone to be patient-specific, meaning that you're recapitulating anatomy, but you're also recapitulating biology. And for the latter one, this means that you will start from the patient's own cells and regrow the bone. The other reason, besides this complexity, anatomical complexity, is that the jawbone is the most loaded bone in our body because of the chewing. So it was very challenging. And we reasoned, if we can make this, we can make pretty much anything else. So we started this project and we studied this over years to understand what are the regulatory factors, like what is the medium composition of the culture medium, how fast we need to perfuse it through the bone, how much oxygen we need, how much mechanical stimulation we need, etc. And we were getting some really, really interesting data. And then one of these studies was published in PNAS, which stands for Proceedings of the National Academy of Sciences. And somehow this article caught attention of New York Times, a journalist covering science. So one day in the Sunday edition of New York Times, we got half a page under title, Human Bones Grown to Order. And this was a very interesting experience that after all these scientific papers and our excitement about the discoveries we were making, there was something that actually turned around everything and was more influential than many of these scientific publications. So people started to calling us, people who are in charge of entrepreneurship in the New York City and economic development and all this, and discussing with us funding of something that National Institutes of Health would not fund at that time within our project, which is this translational effort to show the technology works at a clinical scale. So we got critically needed funding, and we conducted very, very extensive studies in large animal models. So this is a human-sized pig. And we showed in this human-sized pig that the technology works, that after 6 months, these animals really had a new jawbone that was well integrated, functional, etc. So this study was the reason why we were able to go further. So we launched a company called EpiBone that took over the translation of this technology. They conducted some clinical studies of this particular technology and the variation of the technology for other bones in the body and also other joints that also contain bone and cartilage tissue. And so EpiBone is successfully continuing their journey. So the technology has already seen small groups of patients and the process is continuing. And we do hope that this will reach more patients and more indications, more different clinical needs.
Was there a moment in your career when you realized that this kind of bioengineering was possible?
Yeah, I think the aha moment was already in Boston. We started with heart and bone tissues. And somehow people described our lab as a lab that's fixing broken bones and broken hearts, which sounded like very cute. So my aha moment was really with the heart tissue. This was before human stem cells. We were all using just animal cells. And placing these animal cells into different scaffolding materials and stimulating them electrically, I will always remember seeing under the microscope one day this teeny tiny piece of tissue that we made in lab beating. And this looked to me like a miracle that we were able to provide the cells with some minimal set of regulatory cues. And cells are smart. They just figured out the rest and picked up, generating additional signals and formed like a synchronously, regularly beating piece of heart tissue. And given how complex the heart tissue is, it just occurred to me like there are many, many challenges. There are decades of research that are still ahead of us, but it showed it's possible. So there was this potential, that you can make in lab something that is fully biological, that could potentially be used to restore or regenerate tissue in our body that is lost to injury or disease or just never existed because of some kind of congenital abnormality.
I have a specific question about your lab and the bioreactors that you use bioreactors to mimic the human body. What exactly are those machines doing?
These machines are trying to fool the cells to think that they are inside the body by providing them with conditions that are as close as possible to the conditions in the human body. So you have three components in any tissue engineering recipe. The first and most important one are the cells themselves. So these are the stem cells, adult stem cells, in most cases obtained from a very small sample of blood. These cells can become any cell type and assemble into any tissue type in our body. So you need regulation. At regulation, and you need to inform the cells what you want them to become. And this is what we do using two bioengineering tools. The first one is scaffold, the biomaterial. So the cells are placed into this biomaterial that is providing structural template, defining the shape and size and architecture of the tissue, but is also providing signals, chemical signals and also physical signals. Like you can imagine that the scaffold for forming bone will be stiff, and scaffold for forming, say, brain tissue or muscle tissue will be nice and elastic and very, very soft. So this is tool number one. Tool number two is the bioreactor, which is like a substitute body. So you are providing nutrition and oxygen and removing metabolic products. You are bringing in different bioactive molecules that are important. You are also bringing in physical signals such as electrical or mechanical or hydrodynamic, depending on the tissue type. So if you combine these three components the right way, then you can get a piece of functional tissue at very small scale for testing, for studying diseases and testing drugs, or clinical human scale for transplantation for use in clinic.
I'm curious if you or the folks in this lab ever think about the ethics of growing human tissue. Is that something that is ever discussed?
All the time.
Yeah?
All the time. Yes. So, very often, you may have capability to do something, but you're not supposed to do it. It is incredibly important to do certain types of research when we are ready for it. And actually, there are ethical considerations that make us draw a line. So you need to have this red line about how far you can go. This is not only your decision. You really need to understand and find a way to get inputs from patients, from organizations representing patients, to have a full understanding of the workflow that would lead to clinical implementation and any possibility of any adverse effects, of any harm that can be done, physically or psychologically to the patient. So having an informed situation is incredibly important. So for everything we do in laboratory, besides our own ethical concerns, we do need to work under specific protocols that have been written by us, but then reviewed and eventually approved by the institutional review boards. So there are protocols for human subjects. For example, if you are obtaining samples of blood or tissue or saliva or anything from the patient, this has to be tightly regulated. The patient needs to consent to use of their biological material in research, and not in a generic way, but with full understanding what kind of research are you conducting, what is the goal of this research, and what is involved in this research. I mean, you probably know about HeLa cells that were used completely illegally from a Black woman many decades ago. This is not something that can happen today. In addition to human subject protocols, there are also animal protocols, because we are trying to minimize the use of animals in research, to use them only when you absolutely need the whole organism environment and there is no other option to do it. But then again, you work under the oversight with the protocols that are approved and with oversight of another board, which is for animal studies. So there is a lot of regulation, which is great because your own judgment also depends on your experience, on your own moral principles. So I think having these bodies that are regulating what we can and cannot do is really critical for doing things right.
Have those bodies kind of remain consistent throughout your career?
Yes, they are. Each institution has, they are also governed by some broader guidelines, like within this country, for example, but each institution would have their own boards, and they are there to help you to develop the right protocols and also to follow these protocols. If your science is bringing you to the point that you realize something and that would actually make some modifications of the protocol very beneficial, you cannot just embark on those. You need to go back to the board, justify requested protocol modification, and get approval. So there is very tight and very meaningful regulation of both animal and human tissue work.
In your mind, what are the hardest problems in your domain that are the most ripe for breakthrough in the next 5 to 10 years?
I think in principle, what is a huge challenge is biology. It's not engineering, because I believe that if we know what are the conditions that we need to provide to the cells to drive them into specific tissue structure, we will always find a way to do it. So biology is really very challenging and very complex. The good news is that as we are building these models, human tissue models, we also learn about biology, because these models are in many cases very faithfully representing the conditions in the human body. So you make the best model you can based on the knowledge that you have today. And then by experimenting with this model, you get information that helps you build a better model, which gives you more scientific insights. And this is a sort of discovery implementation cycle that is really driving our efforts. So this is maybe across the board in general. Specifically, when you asked about tissue systems, I think that the experience shows that some of our organs are definitely much more difficult to recapitulate in lab. And this really goes into brain, immune system organs, and cardiovascular. I think these are much, much more demanding than the other ones. It's also known that the tissues that are sort of volumetric, not flat, not thin, are much more difficult to make. Some of the first successes in tissue engineering were accomplished with either tissues that are very slow, such as cartilage, so they don't need much, or tissues that are very thin, such as bladder or skin. And then as the field was evolving, then we were moving towards more complex situations. And then today, we also have these models that are like multi-tissue or multi-organ-on-chip models, where you, for example, model the effects of ischemia, or you model a specific cardiomyopathy by connecting different tissues to each other. Like you have heart that you're studying, and then you have a liver that is taking care of metabolism, and you have a bone marrow that is a source of immune cells, etc. And these tissues, if you connect them to talk to each other, they will allow you to do this kind of systemic modeling.
Can you tell us about a project that you're working on now?
There are very many projects. Maybe I can mention two that are like at two different ends of scale.
Yeah.
Some years ago, we developed a technology for maintaining a human lung outside the body for a prolonged period of time. And this project was motivated by the clinical need for more lungs for transplant for people that are at the end stage of lung disease. And the problem, based on what the transplant surgeon told us, was that not only the number of donor lungs is lower than needed, but they need to reject very often 3 out of 5 lungs that are received here because they are deficient. They are not functional enough. So we started to work on how to repair these lungs to meet the standards for transplantation. And to do that, the first step is to maintain them outside the body on ventilation, blood circulation long enough to have time for the repair, for different interventions. So we figured out this technology, which actually resulted in another startup, which is now nearing the point of starting clinical studies in this space. At the same time, we were approached by a foundation that is focused on cystic fibrosis to see if we can use this platform to establish a model of cystic fibrosis that can be used to optimize the delivery of therapeutic. Cystic fibrosis is genetic disease, so if you can diagnose it early enough, which is possible today because the mutations have been identified, then you need also efficient delivery, and this is what we are working on. So this is like something that's ongoing at the level of clinically sized organs. The other project that we are working on is the use of organs on chip for modeling different kinds of diseases. And these diseases include cancer metastasis. They also include studies of immune disease-induced cardiomyopathies, myocardial infarction, etc., etc. So it's really going on, and it's based on pretty much the same fundamental foundation, but then each student has their own take on what they want to study.
Well, thank you once again, Professor Vunjak-Novakovic, for joining us on Trailblazers.
It was really nice to talk with you.
Trailblazers is a production of Columbia University's Office of Public Affairs. You can listen to our other conversations on Columbia's Impact and Innovation website, our Vimeo page, or our YouTube channel. Thanks so much for listening.